Good morning, sir.
Good morning, all of you. So, welcome back to online classes. This is Krishna sir, Geology. Today, we are going to continue of antigen antibody reactions. We already discussed what is antigen antibody reactions. Uh, Antigen antibody reactions means the soluble antigens or soluble antibodies bind with the to be formed Padma immunocomplex antigen antibody complex are formed. So these antigens are soluble, antibodies are soluble, both are mixing, both are combined, both are binding to react to be form of antigen antibody complex. The antigen antibody complex are formed. Okay, so in that what happened ma? See, complexes are occurred, clottings are occurred, precipitation should be occurred. So different so absonation will occur. So this totally different complexes are we are occur. So these complexes are called immunocomplexes. So what are the antigen antibody reactions? Right, mainly remembering one thing, the antigen antibody reactions. Number one, precipitation. So number two, agglutination. Number three, complement fixation. Number four, ELISA. Okay, and next absonization, neutralization. So many reactions are there. It will be asking some that means in entrance level like CSIR-NET level and entrance level. So we'll asking compulsory one question from that these antigen antibody reactions. So in these antigen antibody reactions, antigen binding site is called epitope, antibody binding site is called paratope. So both are fixing with the lock and key fit theory. With the lock and key fit theory, right? So same phenomena we are discussed. So by using of remembering one thing, these antigen antibody reactions are called immunological techniques. Immunological techniques, nothing but in the serological techniques. It is also called serological techniques. It is also called immunological techniques means by using of these techniques to identify the disease. To identify the disease like typhoid, like rheumatoid arthritis, like identify blood clotting tests, so like etc. For example, the precipitation already discussed. The precipitation test we already know very well. This precipitation, which is helpful for identification of the determination of SCG protein, that pregnancy test, and what one Felis test, like different tests we are knowing. Then agglutination. What is precipitation? Precipitation means soluble antigens are bound with soluble antibodies to be form of insoluble antigen antibody complexes in the tube. The precipitate should be occurred. That is called what one precipitation. Crystal precipitates are occurred in the sample. And agglutination, okay, soluble antigens are bound with soluble antibodies to be formed. Soluble, insoluble antigen antibody complexes are like clamps, blood clots should occur. By using of these blood clotting, the blood clotting that is agglutination test. This agglutination test, okay. So by using of this agglutination test, mainly two types are there: active agglutination and passive agglutination. Test tube agglutination or slide agglutination. Tube agglutination test by using of tube viral test we are going to conduct, and slide agglutination also rapid agglutination, rapid viral test we are going to. That means identify of salmonella typhi. That means typhoid fever we are observed. So this viral test useful for what ma? So that is very well identify, that means diagnosis of typhoid fever, and this rapid agglutination by using of this test, this immunological test to identification of the blood grouping. So already told according to Karl Landsteiner. So what ma? By using of this agglutination test, he was discovered ABO group system. ABO nothing but A group, B group, AB group, and O group also. Right. So this is a very important thing. Next, that means passive agglutination. This passive agglutination test, okay. By using of this passive agglutination test, to identify of rheumatoid arthritis. Rheumatoid arthritis. So these are the tests we are earlier class we completed. Now today we are going to discussing about complement fixation. Complement fixation. So medible visible ma Priya, Divya, Sharada.
Yes, sir.
Okay, thank you. So the complement fixation, the lysis of RBC. What is a complement fixation? Simply one. So whether antigen antibody complexes are formed in that the case in the blood, complements are activated. Complement are activated. Whether antigen antibody reactions are formed in that complements are attached with bind with the these antigen immunocomplex or antigen antibody complexes, the reaction will give very fast and phagocytosis, abstination, similar different reactions will start. So this complement fixation, lysis of RBC or bacteria require some non-specific unstable components of fresh serum which are called complement. What is complement? Lysis of RBC, bacteria require some non-specific unstable components of fresh serum which are called what ma? Complement. This complement system comprise how many proteins? 11 proteins are there. 11 proteins are present even individual. They bind fragment of complement site of antibody. They bind to FC component, that means FT component of FC component of AB involved in antigen antibody complexes. This ability of antigen antibody complexes to fix complement used to complement fixation test. Just now I told you. So whether antigen antibody bind, for example, this antibody binding with antigen, that is paratope with help of FAB, that means fragment of antigen binding site, FAB. So this is called FC. Where the antigen antibody complexes are formed, bind with the antigen, then this complement are attached to FC site and to be start complement fixation test. Okay, right. In the first stage, the test antigen and the antiserum nearly approximately heated 56 degrees Celsius inactive complement. First of all, to inactive the complement and mixed in the presence of known. The mixed in the presence of known about complement. This incubated 4 degrees Celsius for 18 hours. Then, if AB specific for the AG is present in the serum, antibody specific for the antigen is present in the serum. The antigen antibody complex will be formed that will be fixed the complement. Okay, for example, okay, you know very well. Okay, in the complement fixation test. Okay, see here, antibodies are there, serum outlet antibodies are there. So with the antigen with the antibodies, these antigens, these are the antibodies are there. So and with complement, these are the pink color is called complement, right? These are the antigen. These are the antibodies. These are the antigens. Both are mixing the serum. Here, these are the antibodies. These are the antibodies. These are the black color spot are called antigen, and Y shape that is antibodies. These antigen antibody complex are formed. Then, when the antigen antibody complex are formed, these complement are fixed. The complements are fixed. Okay. In that the case, totally to sensitize what happened. So these antigen antibody complexes will give the reactions. These complement are to kill the bacteria and RBC membrane, to lysis the RBC membrane and bacterium cells are there. So by using of this complement fixation, that is what phagocytosis reaction takes place to kill the microbes with the help of this complement fixation. Next one, ELISA. ELISA means E means enzyme, L means linked, I means immuno, S means sorbent, A means assay. What is ELISA? ELISA means, what is ma? So ELISA, enzyme linked immunosorbent assay. E for enzyme, L for linked, I for immuno, S for sorbent, A for assay. Enzyme linked immunosorbent assay. So this enzyme linked immunosorbent assay, it is also one of the immunological test. It is also one of the antigen-antibody reaction. It was discovered by Pierleman and Angwell. What ma? Pierleman and Angwell scientist. Pierleman and Angwell scientist was discovered this. What is that ma? This ELISA test. In 1971, it was discovered. In 1971, enzyme-labeled antigens, antibodies were developed in serological reagents for the assay of antibody and antigens. So, the very simple, sensitive, and economic and less hazard when compared to RIA. So when compared to radial immunoassay, it is very easy, very sensitive, very economical, less hazard. So here, what happened? The ligands are used here. The ligands are used here. A molecule which can detect the antibody is covalently coupled with enzyme such as the peroxidase, beta galactosidase, alkaline phosphatase, different enzymes we are using. Simply here, that is coated antibodies, we are enzyme coated antibodies we are taking. Enzyme coated antibodies we are taking. Okay, and so different we are going to, that means by using of this ELISA, identification of antigens, diagnosing of antigens, diagnosing of antibodies. Okay, identify the purify the antigens, purify the antibodies, purify the antigen antibody reactions, antibody antigen antibody process, immunological by using of this test. Okay, not only this ELISA, the ELISA test actually nowadays using for to identification of the HIV virus, to diagnose the HIV virus, to identify the AIDS disease. But not only in this ELISA, not only to identify the that means HIV AIDS, but it is also useful for identify the Plasmodium vivax, identify the so many viral diseases in the plants. Okay. So this is the, and to purify the antigens, purify the antibodies, etc., etc. So like here we are using the enzymes. What are the enzymes we are using? Peroxidase, beta galactosidase, alkaline phosphatase, etc. we are using here. ELISA is how many types? ELISA is how many types? ELISA is a 3 types of there. One is indirect ELISA, second one sandwich ELISA, third one, what is that one? So these are the competitive ELISA there. Indirect, sandwich, and competitive ELISA there. What are those here? Okay, now let us go to indirect ELISA. What is it, ma? What is indirect ELISA? Okay, so indirect, on the name indirect, so indirectly we should identify the antigens. Okay, this technique used for the detection of the HIV, identify the HIV virus, diagnose the HIV virus. The envelope proteins are developed by a recombinant technology and coated on the surface of the microtubule plates. Suspect the serum is added and unbound proteins are washed. For example, indirectly, we should be identify the antigens by using of coated antibodies, enzyme coated antibodies, right? Very simply, for example, antigens are there in the tube. For example, the blood sample, we should add the antibodies. Okay, here antigen antibody complex are formed. We wash it. Is there any unwanted material is there? Unbounded material to be washed out. Then add antibody coated with enzymes. That's what happened. Antigen antibody, enzyme antibody complex are formed. Okay. Then the enzymes add with the dyes. It will be give the colors. If the positive, if the antigens are there, it gives the positive result. If they're not there, negative result will come. For example, see, and we'll explain later one by one. The sandwich model. Here, what happened? Use to detect the presence of antigen in a sample. The well, that means the well is coated with antibody specific to antigen, and then suspect serum is added in order to react. The wells are washed to remove unbound. For example, here what happened, ma'am? Antibody. So here, uh, the wells are coated. For example, according to 1971, according to according to Pierleman-Angwell scientist, so he was discovered the 96 wells plate are there, ELISA plate. The ELISA plate which coated with polystyrene, with 96 wells are there now. So that is using for the indirect ELISA method and sandwich ELISA method and competitive ELISA method, right? In the sandwich method, first of all, in the indirect method, first coated with antigen, second antibody, third enzyme coated antibodies are formed. It's totally antigen, antibody, enzyme coated antibody complex to identify the ELISA indirectly. But sandwich model, what happened now? First of all, the wells are coated with antibodies, then antigen, and then suspected serum to be added, right? Antibody, antigen, antibody. So totally between the two antibodies, antigens are there. Between the two antibodies, antigens are. That is what ma sandwich. This is what ma sandwich. Sandwich is what we are eating like pizza, burger. Above the bun is there, below the bun is there, right? Between the jam we are taking. That is called sandwich. Same phenomena here also, right? I think catch my point. Used to detect the presence of antigen in sample. The well is coated with antibody specific to the antigen, and then suspect the serum is added to the well. Or for example, if it is if the antibody coated antigens are added, antigen-antibody complex are formed. So then, with the help of the water, remove that one. Okay, only complex are there. Then again add with what ma antibody coated antibodies, enzyme coated antibodies. So the antibody, antigen, antigen. So totally antibody, antigen, antibody complex are formed. Right. So here with help of removal with help of the water, distilled water, so unbounded particles. Right. This is called sandwich ELISA. For example, in the indirect ELISA we are taking here. See, simple phenomena. First of all, the wells are coated with the antigen. So in the indirect verma, the wells are coated with antigen. Okay, red spots are there. And wash with the help of the distilled water. Because of here, the polystyrene plates are attached with the own antigens. The unbounded particles removed with the help of the water. Then we should add suspected serum, right? And specific antibody to bond with the antigen to be formed for antigen-antibody complexes. See here in the tube, only antigen-antibody. So here again wash unbounded particles to remove from that solution. Then, okay, to be formed and then add, what happened, ma'am? Enzyme coated antibodies. What, ma'am? Enzyme coated antibodies. So here add enzyme conjugated secondary antibodies will be added. Totally what happened, ma'am? Enzyme coated antibodies like antigen, antibody, enzyme coated antibody complex are formed. So here wash with the help of the distilled water any unbounded particles. Then in the substrate, what happened, ma'am? Measure to color, the color to identify if the antigens are there. The patient sample, patient serum which contains antibodies. Okay, easily we should identify. For example, nowadays RT-PCR test. RT-PCR test useful for what, ma? What is RT-PCR test, ma? RT-PCR test is also one of the important tests for one type of color intensity. It gives the colors. So that is called indirectly. Indirectly, we should identify the patient disease, right? That is indirect ELISA by using the indirect ELISA to identify the HIV virus, Those are the people suffering from the AIDS disease. The second, sandwich ELISA. What is that, ma? Second one, sandwich ELISA. The sandwich ELISA, remember one thing. So here, first of all, the well is coated with the antibody. The well is coated with the antibody. Then with the help of the water removal. Okay. Then add antigens. Simply here also antigen antibody complex are formed. Then with the help of the distilled water remove that sample unbounded particles from the tube. Then we should add conjugated enzyme coated antibody we are add. So here what happened, ma? Antibody, antigen, then enzyme coated antibody are there. So in this what happened, ma? Then again we should add substrate. We should add the substrate, then the color will be formed. The color will be formed. So add the substrate to measure the colors. Whether the adding the substrate, so what happened, ma? The color will be formed, that is give the positive result. This is called what, ma? Sandwich ELISA model. Sandwich ELISA model, right? Now let us competitive ELISA. Competitive ELISA. In the competitive ELISA, remember one thing. Okay, what is that? So the competitive ELISA incubate the antibody with the antigen to be measured. Yes, separate. For example, the antigen antibody complex are incubated first of all. Incubated in the what happened, ma? So in incubator, right? Then these antigen antibody complex are mixed to antigen coated well. Antigen coated well. So antigen antibody complex are incubate and then add, okay, so mix it to antigen coated well. In that the case, what happened? Wash with the help of the distilled water. So unbounded particles. Here then add, what happened, ma? Enzyme coated, conjugated enzyme coated antibody added. So here what happened, ma? Simply remember one thing. Antibody, enzyme coated antibody, and next antigen antibody complexes. Okay, so with so totally here complex it is looking like an indirect method, but indirectly, directly we are already incubated fixed with antigen antibody complexes and stored. That's why. So then again add with the substrate and it will be color will be formed. So the result will be positive. If the color should not form, the result will be negative. So this is what very important thing. And see, these are the important. So in the competitive ELISA, I already told you. Another variation for the measuring amount of antigen competitive ELISA. In this technique, antibody is first incubated in solution with a sample containing antigen. Then antigen antibody mixture is added to an antigen coated micro well. This micro antigen present in the sample, the less free antibody will be available and unbounded particles were removed with the help of the distilled water. The less free antibody will be available to find the antigen coated well. Addition of enzyme conjugated secondary antibody specific for the isotype of primary antibody can be used to determine the amount of primary antibody bound to the well as in the indirect ELISA. Simply, it is also one of the similarly indirect ELISA method. Okay, so these are the totally ELISA. So very important. So this ELISA, very important. This ELISA test in 1971 prepared by Pearlman and Angwell in 1971. So the well which is coated with polystyrene, the wells are this method with the how many wells? 96 wells are there totally, and so this ELISA method to useful for the identification of the HIV virus or lab diagnosis of AIDS disease. Okay, and to identify the antigens, to identify the antibodies, to purify the antigens, to purify the antibodies, and to identify the antigens in the that means not only animals, it is also to identify the viral microbes also. Okay, that is clear. Done. Now let us go to next important antigen antibody reactions, that is immunological techniques. Immunological technique, right? So right, you know very well. See this, that is what immunofluorescence. What is that, ma? Immunofluorescence. What is immunofluorescence? Immunofluorescence means immuno. That means immuno means antigen antibody. Immuno means what, ma? Antigen antibody. We should add the fluorescent dyes. Simply. So immunofluorescence, in the sense, okay. So this immunofluorescence is the property of absorbing light rays on the particular wavelength to emitting rays with a different wavelength. Fluorescent dye shows fluorescent dye shows are brightly under UV light as they convert into visible light. Simply, the antigen antibody complex are coated with simple fluorescent dyes. Right, the fluorescent dyes with the light emission takes place, it gives the colors, visible colors, the positive result will be found. Right. This technique was the Kuhn et al. 1942 showed that labeled dyes can be conjugated to antibody. These labeled antibodies can be used to detect antigens. So first of all, these antibodies are coated with dyes, the fluorescein, rhodocin, so whatever it may be, right? The dyes are the commonly used in including, that is what fluorescent. Fluorescein in organic dye. The fluorescein in organic dye that is most widely used a label for immunofluorescence. The fluorescein is an organic dye that is the most widely used label for immunofluorescence. In the immunofluorescence, we are using the fluorescent dye for the identification of the antigens. Okay. The procedure observed the blue light. Nearly approximately the light wavelength, that is 490 nanometer. How much, ma? 490 nanometer, and emits in the intense yellow-green fluorescence. The blue light to give in 490 nanometer and emits intense yellow-green fluorescence in 570 nanometer also. One more dye also we are taking. What, ma? Phycoerythrin. Phycoerythrin is an efficient absorber. Phycoerythrin is an efficient absorber of light, nearly 30-fold greater than fluorescence. 30-fold greater than fluorescence. And a brilliant emitter of red fluorescence, stimulating its wide use as a label for immunofluorescence. It is a more than, that means 30-fold greater than fluorescence. That's why nowadays we are using the immunofluorescence. Okay, this phycoerythrin we are using, right? Okay, see here, first of all, what are the procedure here? Direct immunofluorescence, right? First of all, the cell with membrane antibody, the membrane antigens, okay? Primary antibody, membrane antibodies are there. These antibody bind with antigen to be form of antigen-antibody complexes. These primary antibodies are there. See, these antigen-antibody complexes are primary antibody. And what, ma? See, the secondary antibody coated with fluorescein, right? In the direct method, this is the direct method. The direct method, the fluorochrome is coated with antibody with them. For example, we are taking this antigen. Here antibody coated, primary antibodies are coated. See here, fluorescein coated, and it is to identify the antigen directly. So indirect method with the fluorochrome, first of all antigen taking antibody, then again fluorescein coated antibody, looking like it is ELISA, looking like it is ELISA. Indirect method, the fluorochrome labeled isotope antibody we are taking for identification. And indirect method with the fluorochrome labeled protein A, so that is what happened, it is indirect method we are using, right? So in this, this is one of the important reactions nowadays. What is the conclusions of antibody antigen reactions? There, thereby we conclude of the topic with a quick review the antigen antibody reactions. The antigen and the antibodies combined specially with each other to be form interaction between them is called antigen antibody reactions. Simply, the soluble antibodies and bind with soluble antibodies and soluble antibodies bind soluble antigens to be form of insoluble antibody-antibody complex, antigen-antibody complexes. So, right. So this antigen and these antigen and antibodies combined especially with each other. This interaction between them is called antigen-antibody reaction. As per the current requirement of the syllabus, the topic has been included right from future till the types of reactions. We show all gratitude towards all of you. Okay, hope the project that means information like what agglutination, the precipitation, agglutination, passive agglutination, and tube agglutination, and ELISA test, immunofluorescence test, okay, RIA, radial immuno test, so and single, double, and circular. So what this is the total important of what this what is that immuno uh, and immunological techniques also. Sometimes we'll asking what is immunological techniques. Sometime what sometime remembering one thing. Sometime immunological techniques also they are asking. Okay, immunological techniques. What are the immunological techniques? We are discussing what are the immunological techniques we are going to see, right? Immunological techniques are word. By using of immunological techniques also known as indirectly anti-antibody actions. By using of immunological techniques to identify the diseases like precipitation test, agglutination test, okay, and the agglutination, precipitation to identify the Felis test, black bone fever, and the pregnancy. And by using of these serological techniques like agglutination, passive agglutination, by using of agglutination to identify the typhus fever, typhoid fever, and by using of tube, that means viral test, we are identify the viral test by diagnosis the typhoid fever, and passive augmentation to identify the rheumatoid arthritis, right? And that means ELISA useful for the HIV virus and AIDS, and immunofluorescence also to identify the different serological diseases by using of the serological test of immunofluorescence, right? And these are the tests useful for the identification of the disease, right? You know very well. Okay, this is one of the important topic for so in immunological techniques will asking one question, and sometimes we'll asking antigen-antibody reactions. Okay, so this is the very important topics of immunological techniques and antigen-antibody reactions. Okay, this is a today topic, and tomorrow also like sharp 7 o'clock I will take the class. Okay, and hypersensitivity, don't miss it, hypersensitivity. Okay, so today, so you must be and all of you get ready for the. Thank you very much, my dear legends. This is Krishna sir, so signing out.